The central dogma is the process by which the instructions in DNA are converted into a functional product. It was first proposed in 1958 by Francis Crick, discoverer of the structure of DNA. The central dogma of molecular biology explains the flow of genetic information from DNA to RNA to make a functional product, a protein. The central dogma states that the pattern of information that occurs most frequently in our cells is from existing DNA to make new DNA, DNA replication, from DNA to make new RNA, transcription, from RNA to make new proteins, translation. DNA stands for deoxyribonucleic acid. This chemical substance is present in the nucleus of all cells in all living organisms. DNA controls all the chemical changes which take place in cells. DNA consists of two molecules that are arranged into a ladder-like structure called a double helix. DNA is a nucleic acid. Like other nucleic acids, it is composed of smaller molecules called nucleotides. Each nucleotide consists of a phosphate group, pentose sugar, and nitrogenous base. The phosphate and sugar form the backbone of the DNA molecule, whereas the bases form the rungs. A DNA nucleotide can have one of the four possible bases: adenine, thymine, guanine, or cytosine. Therefore, there are four possible DNA nucleotides, each with a different base. A DNA molecule has two strands of nucleotides. The two strands are antiparallel, which means they run in opposite directions. Phosphate is found at the 5' end of a strand, and deoxyribose is found at the 3' end of a strand. Two strands are held together by hydrogen bonds between complementary bases. Adenine bonds to thymine and guanine bonds to cytosine. Adenine forms 2 hydrogen bonds with thymine, whereas guanine forms 3 hydrogen bonds with cytosine. DNA has the ability to make copies of itself by the process of DNA replication. DNA replication is semi-conservative. Semi-conservative replication is the process in which the original strands of DNA remain intact and act as templates for the synthesis of duplicate strands of DNA. The new strand is replicated from the 5' to the 3' end. The enzymes involved in DNA replication are helicase, which unwinds the DNA, primase, which attracts complementary bases to form a primer sequence, DNA polymerase, which adds bases to the primer strand by reading the code, and DNA ligase, which seals the sugar-phosphate backbone back together. DNA replication starts with helicase unwinding the strand at the replication fork. DNA polymerase III is the main enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to a 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, which is a sequence of about 10 nucleotides which are complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA.
Because DNA polymers can only add nucleotides in the 5' to 3' direction for the lagging strand, nucleotides are appended fragments by the name of Okazaki fragments.
When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. As an error-checking mechanism, when incorrect nucleotides are added, nucleases eject them. The leading strand elongates towards the replication fork by adding nucleotides continuously to its growing 3' end. Finally, DNA ligase joins the Okazaki fragments together and seals the other nicks in the sugar-phosphate backbone. Transcription is a process by which genetic information encoded in DNA is copied onto messenger RNA. Transcription occurs in the nucleus. The main differences between DNA and RNA is that the sugar in RNA is ribose instead of deoxyribose. RNA is single-stranded. RNA contains uracil instead of thymine. RNA is disposable. RNA can be outside the nucleus, DNA cannot. There are 3 main types of RNA: mRNA, or messenger RNA, encodes the amino acid sequence of a polypeptide. tRNA, or transfer RNA, brings amino acids to ribosomes during the translation of RNA to protein. rRNA, or ribosomal RNA, along with ribosomal proteins, make up the ribosomes, the organelles that translate the mRNA. There are 3 steps in the transcription of DNA to RNA. Initiation. In initiation, RNA polymerase recognizes the promoter and starts the transcription. Elongation. In elongation, the RNA strand is continuously growing. Termination. In termination, the RNA polymerase stops synthesis and the nascent RNA is separated from the DNA template. Eukaryotic genes coding for different types of RNA are interrupted by unrelated regions called introns. The introns must be removed from RNA before translation in a process called splicing. Other parts of the gene surrounding the introns are called exons. Exons contain the sequences that finally appear in a mature RNA product.
Unlike DNA replication, transcription does not involve the use of RNA primers. In addition, transcription begins with initiation of the promoter region, which shows the upstreams of DNA sequence to be transcribed by the RNA polymerase. The process called elongation happens where RNA polymerase adds matching RNA nucleotides, which are paired with complementary DNA nucleotides of the coding DNA strand. Furthermore, sugar phosphate backbone forms the non-coding strand of the DNA, which is left alone.
RNA processing is a process in eukaryotic cells where primary transcript RNA is converted into mature RNA. Mature RNA is a eukaryotic RNA transcript that has been spliced and processed and is ready for translation in the course of protein synthesis. The first step in RNA processing is capping with the addition of a modified guanylic acid to a 5' end. Second, adenylation occurs with the addition of an adenine tail to a 3' end. A gene consists of both introns, regions not represented in the product, and exons, regions that encode part of the protein product. The introns are transcribed with the exons, however they are spliced out in the third step of RNA processing. Ribonucleic assemblies, called spliceosomes, perform the splicing. The result of these steps is an mRNA. DNA translation is the process by which information encoded in mRNA is used to assemble a protein at a ribosome. DNA translation occurs on a ribosome. The actual information for making proteins is called the genetic code. The genetic code is based on codons, sequences of 3 bases that instruct for the addition of a particular amino acid or a stop. Codons are thus read in sequences of 3 bases on mRNA, sometimes called the triplet code. Codons are always written in the 5' to 3' fashion. 4 base types allow 4 cubed, which equals 64 combinations. Plenty to code for the 20 amino acids typically used to build protein. The anticodon is the matching sequence on the tRNA to the codon on the mRNA. When the codon and the anticodon are a match, the amino acid is added to the growing protein. Transfer RNA, also known as tRNA, brings amino acids to the ribosome so it can build protein. It has anticodons, which are 3 nucleotide sequences, which are complementary to the mRNA codon. Proteins are assembled on ribosomes made up of ribosomal RNA. Ribosomal RNA helps build the actual protein. rRNA puts together the amino acid messages carried by mRNA and decoded by tRNA. Translation has 3 stages: initiation, elongation, and termination. The mRNA moves out of the nucleus into the cytoplasm, where it attaches itself to the ribosome made up of ribosomal RNA and ribosomal proteins. The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA, or mRNA, binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Later transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, also known as tRNA, is called the anticodon, and 3 nucleotides on the mRNA or messenger RNA is called the codon. This ensures the correct sequence of amino acids. The messenger RNA or mRNA passes along the ribosome in short bursts of 3 nucleotides at a time. While this is occurring, the initial tRNA is moved to the E site and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The old tRNA is paired with the previous codon, which is passed to the P site and then to the E site as the amino acid is carried and transferred to the growing amino acid chain. The carboxyl group in one amino acid reacts to the amine group in the other amino acid. This results in a peptide bond. As the ribosome proceeds down the mRNA, a stop codon is encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell. A mutation is a permanent change in the DNA sequence of a gene. Mutations in a gene's DNA sequence can alter the amino acid sequence of the protein encoded by the gene. How does this happen? Like words in a sentence, the DNA sequence of each gene determines the amino acid sequence for each protein it encodes. The DNA sequence is interpreted in groups of 3 nucleotide bases called codons. Each codon specifies a single amino acid in a protein. A frameshift mutation is caused by the addition or deletion of nucleotides, which can change reading frame, resulting in completely different translation reading of codons to be code for different amino acids. A frameshift mutation is potentially the most harmful mutation. A frameshift mutation occurs when a nucleotide is added or deleted from the sequence. Both of these shift the DNA, so it changes how the codons are read. The addition or deletion of one or more bases changes the meaning of the whole protein. A substitution mutation occurs when an exchange is done between 2 bases, for example, A instead of C. A substitution mutation results in a different codon, which is a different amino acid. An inversion occurs when part of a DNA sequence is reversed back to front. The inversion results in completely different readings. A silent mutation is a mutation that doesn't affect the phenotype and occurs in non-coding regions. Multiple triplets can code for the same amino acid, so if a base gets substituted for one that still makes the same amino acid, the phenotype won't change. To summarize, genes program protein synthesis via the genetic messenger RNA. The molecular chain of command in a cell is DNA to RNA to protein, and this is referred to as the central dogma of biology.